Welcome everyone to today's Sustainable Farming Association's Soil Health Webinar. I am Wayne Munson, the project coordinator for the Keep Cattle in Minnesota project, part of the Sustainable Farming Association, and I am serving as the moderator for the webinar. It's great to have you with us. I'd like to acknowledge our sponsors, the Minnesota NRCS and the Pasture Project at Winrock International. The Pasture Project is also providing the technical support for today's webinar. This webinar is a program of the Keep Cattle in Minnesota project. The mission of this program is to sustain the beef and cattle industries while protecting and enhancing our vast environmental resources. Cattle work well with improving soil health and also fit well in farming systems which utilize cover crops. Grazing seasons can be extended by grazing cover crops, and cattle can be utilized to remove the cover crop residue from fields prior to planting the field crops. And the manure of the grazing animals is applied directly to the fields. Today's webinar functions as a bridge to the SFA's upcoming Midwest Soil Health Summit to be held on February 18th and 19th on 2015 in Alexandria, Minnesota. Both of today's presenters, Dr. Michael Lehman and Dr. Wendy Tahari, are part of the program, as well as the keynote speaker is Gay Brown from Burleigh County, North Dakota, a pioneer in cover crops, Dr. Alan Williams, a champion for the grass-fed beef industry, and two other producers with soil health and cover crop experience, Ian Cunningham from Pipestone County, Minnesota, and Jerry Doan from Burleigh County, North Dakota, as well as John Mesko and Kent Solberg from the SFA staff will also be presenting. Please register soon for this summit. Last year the summit filled up early. Our first presenter is Dr. Michael Lehman. Dr. Lehman is a soil microbiologist with the USDA Agricultural Research Service in Brookings, South Dakota. His research examines the contributions of soil microorganisms to agricultural production systems and how management tactics can enhance these contributions by soil microorganisms. The end goal is to demonstrate and promote agricultural production practices that require less inputs per unit production. Dr. Lehman?
Well, thanks for the introduction. Let's see. I'm also learning how to use the webinar controls. So here we are. Cover cropping, as Wayne mentioned, is a great tool for building soils that'll work for you. And what's working in there is the biology. I'm a microbiologist. I'm going to talk a lot about microbiology. I'm going to give you a little background on it and then illustrate how it can help your production practices, whether you're growing crops or you're looking to feed cattle, and illustrate that contributions of soil microbes using several research examples. So a famous scholar, Stephen Jay Gould, once said, "We live as we've always lived in the age of bacteria," and kind of wonder why he said it. We're all pretty focused on human beings and our everyday lives. It's hard to imagine that there's a more superior organism on the planet, but yet there is. In the next few slides I want to illustrate that for you. In a teaspoon of soil, there's over 1 billion bacteria. In that teaspoon, there are thousands of different species, and I have species in quotes because we're still having trouble figuring out what makes a bacterial species and what doesn't. Most of you are familiar with the national debt. I think it's $17 trillion. If you get someone to give you $1 for every bacteria in that teaspoon of soil, we'll dig up a shovel, and you've just taken care of the national debt. Try and give you an idea of the overwhelming numbers that this invisible world below our feet has. So another famous scholar named the microbes the unseen majority, because in that teaspoon, in addition to the 1 billion bacteria, are millions of fungi, up to several million algae and protozoan, and dozens of nematodes. And when people think of things like nematodes, they think of pathogens. They think of soybean cyst nematode. But in reality, the bad actors, the ones that are pathogenic to us or plants, are a very small proportion of those that are out there. And most of these nematodes and microorganisms are performing beneficial activities that support life on Earth, including us and our crops. Calculations have been done by another scholar named, well, actually, I think it was Stephen Jay Gould, who did these calculations, or maybe it was Whitman, one of the two. But the calculations, if you took all the microbes that live underneath the soil surface, they live in the soil, but they actually live down to up to 2 miles below the surface, we've recovered viable living microorganisms. If you add up all their weight, you take all these tiny little invisible creatures, add up all their weight across the whole globe, and compare that to the weight of all the living things that you can see on top of the ground: grass, cows, insects, people, trees, grass, vegetation, all that. Apparently, the weight of the microorganisms exceeds that of what you can see. That's one of the amazing kind of facts that really drives home to me how much biomass there is out there, and why we are living in the age of bacteria. So I mentioned about enabling life, and that's just a fact. If there's no soil microorganisms, life will cease to exist on Earth. They are the fundamental ecosystem providers. Their role in decomposition is obvious to many, along with the higher organisms that live in the soil or on the soil. Their role in things like nutrient cycling, a lot of producers are familiar with their role, say, in nitrogen fixation or in oxidation of ammonia to nitrate so it can be taken up by the plant. But of course there's numerous other examples. And then these incredible relationships, and I've got a picture of a soybean nodule where these nodules that are part of the plant actually harbor this symbiotic nitrogen fixing bacteria that cracks the incredibly energetic bond between two nitrogen atoms and fixes nitrogen so it can be taken up by the plant. Those that have livestock, ruminant livestock, are familiar with the role of microbes in nutrition for these animals. The role of microbes in influencing pollinator, both the success of the pollinating organisms, whether they find their source of pollen, but also whether they live and die. All this is based on microbes that live inside of these organisms. And then, of course, there's been a lot of literature that's come out and it's seeped into the, I'd say, the mainstream, really, about the influence of microbes in a person's gut and how they can influence things like obesity, but also things like mood. All sorts of interactions that no one had imagined, and we're just beginning to scratch the surface. So in just last year, 2013, the prestigious American Academy of Microbiologists put out this publication, How Microbes Can Feed the World. So we are now getting widespread recognition of this fact by scientists, obviously some segments of the agricultural producers and livestock managers are aware of this. It's now starting to be part of the conventional literature, and research programs are headed in this direction. And I think that's a great sign. It's taken a long time, and here we are, 2013, 2014, a lot to look forward to. The benefits of microbes, most people think of nitrogen fixation, decomposition. Beyond that, they'd be hard-pressed to name a lot of the benefits that soil microbes can have the system in an agricultural system. But they're really wide-ranging, from being antagonistic to potential pathogens and pests, to suppressing weeds by either chemicals or degradation of the weed seeds, producing chemicals that enhance plant growth. Soil structure and aggregation is a big product of having microbes and having a lot of microbes and a diversity of microbes, and of course soil carbon dynamics and nutrient dynamics. And for the rest of the presentation, I'm going to talk a little bit about cover crops, our research involving soil microbes, and how they influence things like soil structure, carbon, nutrient retention, and nutrient availability. When we talk about agricultural systems, conservation agricultural systems, we basically look at three things. One is some form of conservation tillage. Two is diversifying crop rotation. And three is incorporation of cover crops. Those are kind of the basics, the basic foundation for a sustainable system. I would say, you know, there's not a lot of crop rotation in our region. There's some conservation tillage, I'd say a good amount, and cover crops are on the rise. And I think right now where we have the most chance to make an impact is improving the incorporation of cover crops. And cover crops, as you will see, are an incredibly important component of creating a soil that's biologically active and that will work for you. So a very simple comparison between a field that's covered and a field that's not. Black dirt versus something growing green. It's a freebie. Once you've terminated, harvested your annual crop, you know, usually you're talking about 3 or 4 months where you've got something actively growing on the land, and then often a lot of producers have black dirt the rest. Well, all that sun that hits that land is not being captured. You're not taking an opportunity of the free energy, carbon, and nitrogen that is falling on that land. And if you do that, the plants and the microbes will take that energy and turn it into more carbon and nitrogen in your soils. By doing so, they'll create better water infiltration, better storage, and reduce water erosion and reduce wind erosion. There's really not a lot of controversy about these kind of things. This is one way to jumpstart your system, it also benefits your livestock. Whether you're talking about large organisms like cows that you can see that can forage on the material, wildlife, and some economically important wildlife in this part of the country, pheasants and waterfowl, but also insects, particularly beneficial insects and pollinating organisms. They really are going to be in lower numbers, or they're going to be completely absent from a bare soil. Or if you have something growing there, you can have more of these organisms. But what people don't generally understand is the effect of black dirt, bare soil on the microbes. The microbes need food to grow. They need the carbon that's produced by the plant that leaks into the soil to grow. They need the residues. They need the temperature moderated. They need the moisture moderated. They are susceptible to UV light on the surface of the soil. They're, of course, susceptible to mechanical disruption by the plow, and they're susceptible to removal by erosion. So, this is one of the most important slides of the presentation. The take-home message here is 10 to 40% of the carbon that's fixed by a plant is actually exuded from the plant roots into the soil. That's a lot of carbon that's that's exuded into the soil. This arrangement has been going on for hundreds of millions of years. There's obviously a benefit to it from the plant. Why give up 10 to 40% of your hard-won carbon that you fixed from atmospheric CO2? And the benefit they're getting is by stimulating the microbes in the region of the plant roots, also known as the rhizosphere. This 10 to 40% is apart from the carbon once the plant senesces and from the roots and from the above ground biomass that falls on the dirt and decays and gets incorporated. So this is why the plant is living and actively photosynthesizing. So at our lab here in Brookings, South Dakota, we've been working on cover crops for about 15 years. And we've been examining and trying to measure ways that cover crops promote soil biology and things that have a benefit to agricultural production systems. One of the things we've measured, and this gets into another kind of area, when you're talking about soil microbes, you generally can't see them. They all kind of look the same, and it's really hard to tell what they're responsible for. So we struggle to find meaningful measures. And one of the measures of soil microbial community is the ratio of fungi to bacteria. And it has been shown in the literature that generally, as the fungal bacterial ratio is increased, these systems retain more carbon and nitrogen. And those are positive things. We would like the systems to retain as much carbon and nitrogen as possible. We established this replicated study in the year 2000 at our lab. It's all no-till. It's a 2-year corn soybean, and there's 3 levels of residue removal from the corn phase. It's the corn, one of the treatments is the corn is harvested for grain. The next treatment is the corn is harvested for grain, and then the stover is cut and then baled, and I've seen a lot of that this year, a lot of baling of stover. And then the third treatment is simply silage, where you remove all the corn residues for silage. And the reason we started this is because this sort of cellulosic ethanol production systems have been on the horizon for a long time, and the concern is taking your residues off the soil every year, or every other year in this case, is going to result in a decline in soil carbon. So there's going to be some economic necessity in balancing, whether it be selling the bales for ethanol or using it to mix with distiller grains for feed or as bedding, one thing or another. There's evidently a reason to start baling more corn stover, because I've seen it this fall. And so what we did in 2005 was institute a secondary treatment where we've added cover crops to these corn soybean plots in an effort to try and offset potential negative effects of the corn residue removal. So here's just typical what they look like if you take the grain only. Again, no-till. If you bale the stover, or if you take the silage, you have big difference in what the surface looks like. And then we looked at fungal bacterial ratios, and that's what this graph shows. And low residue removal, LRR, is where we just take the grain. Medium is where we bale. And HRR, high residue removal, is stover. This is after about 12 years, so 6 residue removal cycles. We looked at fungal bacterial ratios. The darker bar has no cover crop. So if you just look at the darker bar, as you remove the residue, you have declining fungal bacterial ratios. It suggests a change in the soil microbial community, a change that is going to go along with less retention of carbon and nitrogen in the system. We also have other data that shows a decline in soil aggregation and an increase in highly erodible fractions with repeated residue removal. On the other hand, the light green bars are the treatments where we added cover crops to. And here you can see that we're able to maintain our fungal bacterial ratios by the addition of cover crops in both the baled stover, the MRR, and the silage treatment. So that's an encouraging role for cover crops. Among their other benefits would be maintaining these, retaining carbon and nitrogen in the soil, and maintaining the erosion resistance of these soils. People usually ask, well, what cover crops? Well, the cover crops changed a lot because in terms of each year, sort of a different set of conditions, whether it be raining or not raining, and different tactics were used to get them established, either broadcasting or if it was an early harvest, direct seeding. So the tactics and the type of cover crops did change over time. And there were some years where we had very little growth of the cover crops because the spigots shut off in the fall and there was no rain. But over a 12-year period, we see an increase in positive soil qualities with the incorporation of cover crops. The other thing we've done is look at how cover crops can help with plant nutrition, specifically nitrogen and phosphorus. When you talk about nitrogen, I mean the microbes are all over nitrogen. I mean they are doing things like fixing nitrogen to ammonia, converting it to organic nitrogen. They're oxidizing ammonia to nitrate. Nitrate is preferentially taken up by plants. They're mineralizing organic nitrogen to ammonia, which can then be oxidized again and be taken up by plants. So they're really the key at increasing the amount of nitrogen that's in the soil and moderating the amount of bioavailable, plant-available nitrogen there is. Phosphorus, they don't directly transform it in these what are called redox transformations, but they can change the form of phosphorus so that it is plant bioavailable. And they do this by producing compounds that can complex P, by producing acid that changes the pH and mobilizes P. They can store phosphorus, and they can mineralize stored phosphorus. And specialized organisms that we'll talk about in a few minutes can also uptake phosphorus and transport it directly to the plant. These are arbuscular mycorrhizal fungi, and their filaments and spores are indicated in this picture in the lower right-hand corner. Okay, so we're first going to talk about cover crop nitrogen mineralization. We have 4 treatments here. One is clover, fallow with no cover crop. We have cereal rye. And we have hairy vetch. And it was a soybean small grains cover crop. Cover crops terminated and then planted into corn. And we looked at nitrogen mineralization using an in situ technique, starting with the termination of the cover crop and following that throughout the corn growing season. And the 3 colored sections of each bar are the periods of time. The blue is, say, the first 3 weeks of the season, and the darker gray is the middle 3 to 5 weeks, and the yellow is the last, say, 3 to 5 weeks. And in this data that we collected, you can see clover really provides a lot of nitrogen over the course of the season compared to no cover crop at all. But also that rye and vetch give you additional nitrogen over the growing season. The important thing to note is that you might look at this and say, hey, clover is much better than rye and vetch. Well, we only follow these during the corn growing season. Rye and vetch have a much higher C to N ratio, and they're probably still releasing nitrogen out there long after the clover has released it. So in terms of timing, long-term building up nitrogen in the soil, rye and vetch are also going to help you, and that wouldn't be reflected in, say, 3 months that we made these measurements. So arbuscular mycorrhizal fungi, I promised I would tell you about these guys and their contributions to phosphorus, nutrition, among other things. And these are fungi that live in the soil. They live there in this form of a spore. There's a little diagrammatic of a spore. And when a plant seed germinates and extends roots, they signal the spores. The spores come, infect the roots, and actually grow inside and create this structure here called an arbuscle, which is blown up once and then blown up a second time. And when you see that picture, what you ought to be thinking about is a lung or maybe a kidney. And what do those do? They act as sites of exchange. For instance, your lung is where the oxygen is exchanged for CO2 into your blood, from the air to your blood, and that's exactly what happens in these arbuscles. The mycorrhizae uptake water and nutrients from the surrounding soil. They bring it in through their filaments to the arbuscle, exchange it to the plant, but the plant gives them something back. They give him photosynthate. They give him carbohydrate molecules that that plant has made, and so it's a trade. And the arbuscle is the site of the trade. It's where the deal happens. So this is a very specific relationship. These mycorrhizae cannot grow without a plant host. And that's a very important concept if you're talking about using cover crops and reducing seasonal fallow with cover crops. You've now provided a host for mycorrhizae, so you're more likely to produce more mycorrhizae in your soils. So another thing on mycorrhizae, again, this is really a key picture here. On the left is a small plant without mycorrhizae, and it shows the volume of soil that the roots explore and can take up water and nutrients. And on the right is a seedling that is colonized by mycorrhizae, all those different colored threads, and you can see the difference in the volume of soil that can be explored to take up water and take up nutrients. But it's not just the overall volume. The mycorrhizal filaments are very small. They can access smaller pores. They have a very high surface area to volume ratio, and they have particular capabilities such as the production of enzymes and complexing molecules called chelators and other compounds that change the pH. All these things contribute to the ability of the mycorrhizae to uptake nutrients that the plant can't take up on its own and deliver them to the plant through the arbuscle. So it's not just nutrient uptake, but drought resistance, and then they also have very strong roles in pest and pathogen resistance, because the mycorrhizae is dependent on the host plant to live and reproduce. So it doesn't want to have the host plant be injured by pests and pathogens. So there's a mutual interest there. So what you might not think right off the bat, you look at these top pictures, examples of wind and water erosion, is how something so small can affect these large landscape visuals of the effects of erosion. But that's exactly what they do. When an aggregate is formed, in part it's formed around the bodies of microorganisms, both bacteria, but particularly the filaments of fungi that help bind these aggregates together. On the right, there's a photo of a mycorrhizal filament, a root, and the mycorrhizal spores and filaments, and then this sort of glycoprotein exudates called glomalin, and this behaves as a glue, and it helps maintain the stability of these individual aggregates, which then translate to landscape scale effects in terms of resisting erosion. So, in short, these microscopic organisms enhance soil aggregation, which promotes overall soil structure, which then gets you two very desirable properties. One is water infiltration, so it prevents runoff, sheet runoff, and also storage so the soils can maintain a structure, still store water, and not be saturated or prone to further water erosion. This is the classic big plant, little plant. Dr. Wendy Tahari was working with me here for several years, and we ran this experiment. And on your left is a few sorghum plants with mycorrhizae. On the right are the same sorghum seeds without mycorrhizae. You can see the ones on the left are bigger. You can look at the graph and see their dry weight's bigger. What is most important about this figure is the mycorrhizae that we put in here, we actually collected from a producer's field. This is a producer that has a lot of cattle. He runs a very diverse rotation. He does no-till. He uses cover crops. And we collected the mycorrhizae specifically from that field that we added to stimulate the growth of these sorghum seedlings. So, in general, arbuscular mycorrhizal fungi, or AMF, are nearly eliminated in ag soils. Things that eliminate them include tillage, because they get mechanically disrupted, seasonal fallow, simply there's no host, annual fallow, simple rotations, they are inhibited by high levels of inorganic fertilizer application. There is some data that suggests fungicide applications may inhibit them, and soil compaction, flooding. They're aerobic organisms just like us. They need oxygen. So if we want our soils to work for us, we need to create better conditions for organisms that mycorrhizal fungi, and we can get these benefits. And in particular, our research is mostly focused on no-till situations and reduction of seasonal fallow with cover cropping. So, a lot of producers have stopped by our lab, and one of the questions that comes up is, hey, can you measure these in my soil? Well, it turns out measuring mycorrhizal fungi is very difficult, much like most of the other soil microorganisms. To get a good, really some good data on how many are there, are they important in the system or not, and we measured the number of mycorrhizae in some cover cropping systems using this very laborious technique where we collect soils, we dilute them out, we incubate them with a suitable host plant, and we harvest them, clean their roots, chemically treat them, stain them, wash them. Very arduous technique. And we did about 1,500 of these things. And after that, we were able, we were able to produce this graph. One graph from about, 1,500 to 2,000 of those samples in that previous slide. And what this graph shows you, we have a 3-year rotation, soybeans, small grains, cover crop. Here's where we're taking the measurement. Cover crop gets killed in the spring, replanted to corn. We're just taking a snapshot of how many mycorrhizal fungal propagules are in the soil. And by propagule, we mean a mycorrhizae that is capable of causing an infection in a new host plant. As you can see, the cover crops across the board, we had canola, and canola is actually a non-host. It's a cover crop that does not host mycorrhizae. Oats, vetch, and then various combinations compared to no cover crop. This is a 3-year study, and across the board, the cover crops generally increase the mycorrhizal properties. We found a lot of success with oats. We also found very good success with these mixtures. We also went to a producer's farm and just grabbed a couple samples, well, more than a couple samples. We actually, this was the same producer where we got the stuff, the mycorrhizae for that sorghum slide. And we looked at 6 fields, paired and unpaired, where he had cover crops and not, and he used a mixture, cowpea, winter pea, millet, turnip, radish. And we found the highest number of propagules on his farm as anywhere we found. And 2, we found that the cover crops had double the number of mycorrhizal propagules as no cover crops. So building soil biota really comes down to some really just a couple of principles. It doesn't really take much. You got to provide food, and by providing food, you got to have diversity and as continuous cover as possible. You have to have a habitat, which would be stable aggregates, and a diversity of plants to produce a diversity of niches. It's really not that difficult. It's very difficult to measure and do the science, but in terms of managing, it's actually fairly straightforward if you can just consider these couple key determinants of whether you're going to build soil biota or deplete it. So my vision is to move away from a system I call pass-through, where we're adding nutrients and pest protection and everything that you need to take your crop off, and assuming that a bunch of it's going to be lost from the system or fixed in the system, unavailable, to one that's more self-sustaining so that you can retain the nutrients in the soil, recycle them, but still maintain your yield. The challenge is designing these systems in the current agricultural, production climate so that the economics of them balance out, and that is a big challenge. But we're fortunate that we have examples of people who've done this. We have Dan Forgey out by Gettysburg, South Dakota, who has done this. They've taken all these different principles and put them into practice and have an economically successful farming system. We have researchers down at Iowa State who've integrated all these principles into systems and found that if we can just add a bit of diversity, keep the ground covered, add less synthetic agrochemical inputs to actually just tweak the system, we can make systems that are profitable as well as conserve materials. So with that, I'd just like to say the only way we're going to progress is multidisciplinary research. We need not just agronomists or soil microbiologists, but a lot of different types of scientific studies here, economic analysis, and I think you're going to hold your questions till the end, and we'll move on to Wendy's presentation. Thank you.
Yes, thank you, Dr. Lehman, for that informative and well-diagrammed presentation. I think we'll move on to our next presenter, Dr. Wendy Tahari, and we'll discuss the importance of diversity in cropping systems and the role of cover crops to manipulate the microbial community in cropping systems. Dr. Tahari has a PhD in ecology and evolutionary biology. Some of her publications demonstrates that manipulation and selection of AMF communities can increase plant biomass up to 69% compared to resident communities. She has spent time with her postdoc at the USDA, and now she has founded the research company Terra Nimbus, located in Pelham, Georgia, under which she continues to research soil microbiology on behalf of America's farmers. Dr. Tahari?
Thank you. I'm very happy to be here. Today I'm going to discuss diversity and its importance, and part of my goal in today's talk is to help the layman, the layperson, really understand and get a grasp on why diversity is important and what its role is, how it matters. We hear again and again, you need diversity, it's good, but not very many people really understand why it's good. So my talk is formulated in some steps here. I'm going to talk a little bit about mycorrhizal fungi, which Dr. Lehman has already introduced you to. I'm going to explain some of the reasons why they're so important, and then some of the problems that we have in our large ecosystem scale, landscape scale farming systems are suffering some real problems these last few years with drought and erosion, and I'm going to talk a little bit about those problems and ecosystem resiliency. What makes an ecosystem able to recover from drought events, flood events, things like that. And then to understand all of these concepts, you're going to need a little bit of Soil Ecology 101. So I'm going to introduce you to a few key concepts that hopefully will tie all of this together, and then we can move on to questions afterwards. So mycorrhizal fungi are fungi that form a mutually beneficial symbiosis with another organism, and typically we think of a plant. There's only one exception that comes to mind, and there's one group that will associate with bacteria. But in general, we're talking about a relationship between a plant and a fungus when we talk about a mycorrhizal association. There are actually 7 types of mycorrhizal fungi, and only some of them are really very important to farmers. And they come from a variety of different groups of fungi. The ascomycetes, the basidiomycetes, and the zygomycetes all have members that are capable of forming a mycorrhizal relationship. The endomycorrhiza kind of stand out in that that's an entire phylum, an entire phylogenetic category of organisms that are all endomycorrhizals, and they all associate with plants. And as you can see, the different groups, different categories of mycorrhiza associate with different groups of plants. And there's no need to memorize them, just to get the general concept across that you've got your endos and your ectos and on down the list, all the way down to specialists that only associate with orchids, and they're called orchid mycorrhiza. And it just goes on. But it's the endomycorrhiza that associate with most of our crop plants, so we're going to focus on that group. They are technically called the arbuscular mycorrhizal fungi, and Mike introduced you to arbuscules, and you're looking at 2 of them right here inside root. This is the cell, a single cell inside a corn plant root, and these are 2 arbuscules. On Arbor Day, what do? We plant a tree, right? So arbor refers to tree, and scule, arbuscule, refers to tiny, like minuscule. So the word actually means tiny tree because that's sort of what it resembles, although obviously it's functioning on a different scale. And when a plant's roots are filled with these little arbuscules, we call that root colonized. Now, if that root was filled with the hyphae of a pathogenic organism, then it would be infected. But this is not an infection, it's a colonization. The plant actually invites the hyphae into its root system, prepares channels, sends out hormones to attract them after the seed germinates. So the plant encourages its colonization. And just the fact that some of its cells are occupied, if a pathogen wants to get into that plant, it has exclusion because these guys are already there. So that's one of the ways that they help keep diseases out of our plants. These arbuscular mycorrhizal fungi, which are the endomycorrhizae group, do not produce any kind of mushroom or visible structure. They're only visible under the microscope. So the ectos are the ones that create mushrooms and other visible structures. And they generally associate, for the most part, with other groups of plants, and particularly pine trees and nut-bearing trees. Endomycorrhiza associate with 90% of plant families, and 99% of all plant families do have some sort of mycorrhizal association. They aren't all endo, but 90% are. And 74% of those 99 families, are going to be endomycorrhizal associates. Then 8% of plant families will associate with more than one group. They can have both mycorrhizal and non-mycorrhizal species of plants within their group, and some plants will actually associate cross categories and associate with multiple types of mycorrhizae, but that's probably a smaller percentage. Your non-mycorrhizal plant families that are important crops would be in the brassicas and the pigweed family, the amaranths, and those crops include canola, radish, spinach, broccoli. These are all things you're probably familiar with. Quinoa, which isn't really grown everywhere, but a lot of you may have heard of it, and then, of course, beets. And a lot of our agricultural weeds also fall into this family, and they're non-mycorrhizal. When you want to study and learn more of these organisms, it's good to have a little bit of a grip on the vocabulary that you need, and you'll come across some terms like VAM, AMF, hyphae, and mycelium, and so it's good to understand what these mean. VAM is vesicular arbuscular mycorrhizal fungi, and these are vesicles. They're like little bladders, the little containers where they store nutrients. And the early mycorrhizal fungi that were first identified produced vesicles, and it was believed that that was definitive of the group, so they were named VAM. And then later it was discovered that they don't all produce vesicles, but they do all produce arbuscules. So the V was dropped, and it became arbuscular mycorrhizal fungi. So this is the more current term, but if you see the other term, you know that they're referring to the same organisms. Hyphae are the little filaments that go out into the soil and are actually the body of the fungi. Most people, when they think of a fungus, they think of a mushroom, but that's actually just a fruiting structure. That's not the fungus as it lives most of the time in the soil. Fungi are generally in the form of these filamentous strands. And more than one species of mycorrhizal fungi, or arbuscular mycorrhizal fungi, might be colonizing this plant. And if I wanted to identify all the filaments that belong to a single organism and say these are all that guy, which you just couldn't look at and do, but if you could, you would call that a mycelium, a single mycelium. So plants have multiple myceliums because multiple species and different individuals can colonize them. So I'm going to give you sort of the short list, 14 reasons why you should care about AMF. Well, 1, they build soil fertility. 2, they increase soil structure and water holding capacity. They produce a more nutritious product for humans and livestock. They replace harmful chemicals. They can eliminate nutrient runoff and leaching. They increase plant nutrient use efficiency, particularly for phosphorus, which is a limited resource. So we really need to increase our efficient use, and not just efficient, but responsible use of this limited resource. They offer protection from nematodes, plant diseases. They can increase a plant's drought tolerance and its salinity resistance. They can cause earlier flowering, more flowers, fruit, and biomass, which equates to increased yields. And as an added bonus, they sequester carbon in the soil, which when we're moving carbon out of the atmosphere into the soil is really good because we put a little too much up there, and we need to get it stored somewhere. And at the same time that it's storing that carbon in the soil, it's using it to build soil structure, so it's a very beneficial place to store it. Yes, sirree, it'll grow hair, fix your teeth, and make you taller. That's a lot of claims for one organism, isn't it? But the fact is, it's not one organism. It's a community that we're talking about. And look at the diversity. This is what the spores look like. That's actually a little orchid seed, and that's a different kind of fungi. But for most of what you're seeing here are spores of arbuscular mycorrhizal fungi. And you can see just by looking at the spores that there's a lot of diversity within these organisms. And some of these organisms may be superstars at just one of those traits that I mentioned. For instance, this guy might be really great at controlling your nematode population, whereas this guy over here might be a super glomalin producer, and this guy might be really good at combating specific disease, and this one another disease. So you want this whole community. They they all overlap in their functions, but they don't provide equal amounts of each function. So that's why you need diversity. So increasing your AMF diversity requires specific management practices. Reduced phosphorus inputs, because under high phosphorus inputs, plants will say, I've got enough phosphorus, I don't need you. They can't reason that they're going to miss out on all those other benefits. Phosphorus is the key that they make their biological decision on. If I have enough, I don't need as many mycorrhizal fungi, so I'll give them less sugar. And so they won't compete as hard for those microorganisms. So we need to select plant varieties that are responsive to mycorrhizal fungi, because not all of the same plant varieties are equally responsive. And some of our breeding programs are, they occur under high input systems. So we may be inadvertently reducing the ability of our crops to form this relationship. And we want to make sure that doesn't happen, because this is a very important relationship to the health and resiliency of our ecosystems. Reducing soil disturbance, that's equivalent to no-till or reduced till, because you break up those hyphal networks and then they're not there anymore, and the fungi have to work harder to restore themselves. And some of the mycorrhizal species can reproduce from hyphal fragments, but not all of them. And those are the guys that we see in decline in our agricultural systems, the ones that only can reproduce by spores. And if we have fallow and there's no place to keep them going so they can finish their life cycle and build those spores, because we've suddenly taken away their host plant by harvesting everything, we start to lose those species. And then rotations and cover crops are also very good, because you need a variety above ground to get variety below ground. Now let's take a look at drought, erosion, and ecosystem resiliency. It was Franklin D. Roosevelt who said, "The nation that destroys its soils destroys itself." And this comment was inspired, unfortunately, by the Great Dust Bowl. And in this map, you can see that the effects of the Dust Bowl were most intensive right here in these this dark, deep yellow area. And it was a result of drought, wind, tillage, and other agricultural practices. And you know, a lot of people say tillage. Well, really, you know, but we know tillage was involved because periodic droughts and windstorms have occurred throughout geologic time, and there's no evidence that anything like a dust bowl really began to occur in the Great Plains until we began tilling them. So we changed something critical in the system. During the Dust Bowl, we lost 100 million dryland acres. Up to 75% of the topsoil just blew away. Oklahoma, Texas, Kansas, Colorado, and New Mexico were hit the hardest, those bright yellow area that I showed you in the map. The dust clouds were 10,000 feet in height, and the economic losses were $25 million per day. But the social costs were immeasurable. People literally choked to death on their dust. This is Kansas in 1935. Can you imagine trying to breathe as this came over? Much less if it continued for 2, 3, 4 days. This is Phoenix, 2011. Lubbock, Texas, 2012. Denver, Colorado, 2013. Texas and New Mexico of this year. This dust cloud covered 2 states, and it was over 10,000 feet high. Are you kind of starting to wonder if we really learned our lesson here? You know. So what's happening? Well, erosion is what's happening. And this is an old coal mine, an abandoned coal mine, where I did a lot of my research as a graduate student. That's my advisor and me looking out over there. And laying down here outside of the picture is Keith Vogelsang, and he's taking a picture at these pedons, these little towers of soil that are out here. And they're only a few inches tall. He's very close to them. This is a close-up of them. And they're all over the landscape, these little pedons. And if you look closely and study this picture a minute, you'll see that they all have something in common. There's a little pebble on top of each one, a little cap. And this just shows you how important the physical process of just covering your soil can protect it. I mean, look at how barren this landscape is. Rain, wind, water, it all just swooshes through here. Yet these little towers have survived just because they were topped by little pebbles and they got a little protection. This is the same mine. That other picture was taken back off in this area. And here we have a wall of erosion that's coming down, and the fines, the little fine particles, are creating this plain of coal sand. And this is the edge of the mine, so this stuff is actually off the mine. But what's going on here? We got this little island, this little oasis in the middle of a sea of just fine coal. And if you look, those are my ATV tracks, because I went in there and said, oh, are there fungi here? And I dug them up. And guess what? I did find mycorrhizal fungi associated with these plants. And these hillocks of soil are 1 meter and more. This one is nearly 2 meters high. And this basically is a cover crop that Mother Nature put down that protected that soil. And this demonstrates just how effective that cover crops can be at covering your soil and protecting it. So the take-home message is you want to cover your soil physically, and that cover crops prevent soil erosion. Now let's take a look at what makes an ecosystem resilient. And to really understand the concept, you need to get a grasp on a few terms that are specific to soil ecology. So I'm going to give you a crash course in Ecology 101. We're going to start with the soil food web. Most of you have probably seen this picture or one very similar to it, where you see all the organisms that feed on the other organisms. And when I was a kid and in school, it was a food chain. But as we learned more, we came to realize it was actually a food web. And there's a lot of diversity in soil. It's far more diverse than these diagrams show us. There are so many things just going on. And I'm not even showing you the differences in bacteria because they all kind of look the same under magnification, little dots. So there's a lot of diversity in the bacteria, there's diversity in the fungi, there's diversity in these arthropods, there's diversity in the worms and the nematodes and the protozoa. And some stuff just makes you go, holy cow, what's that? You know? And it's a whole world, it's a whole universe that few people have ever had the opportunity to look at, and it's right under our feet. So diversity is there in the soil. And the take-home message here is that food webs are complex. They're far more complex than we're used to visualizing because we're used to looking at these simplified diagrams. And now I'm going to take the soil food web and I'm going to turn it on its side, literally. And we're going to take a look at it from a different perspective. I'm going to block out the above ground processes, and we're going to just look at the below ground processes. And of course, the last time I did this, someone commented that my birds don't live below ground. So being a smart aleck, I found a bird that burrows. There's a burrowing owl for you. But the point here is that if we look, we can see that we've got this integrated network of guys that eat guys, and at some point if I decide, wow, these big nematodes are really aggressive, they're controlling things I want controlled, I want more of these. How am I going to get them? Well, I could go buy a bucket of nematodes and throw it on my field and hope that they survived in a foreign environment that they've never been exposed to because they were probably grown up in a lab somewhere, and hope that they can compete with what's already there and hope that they might establish in my field. But really, what I need to do is get the ones that I've got something to eat. So I can get these guys, I need more of these. And then I realized, well, to get more of these, I have to get more of these. And if I want more of these, where do I go? You have to leave the soil. And if you note, this is sort of a size thing. You've got the big things, eat the smaller things, eat the smaller things, eat the tiny things. But at the very top of it all is the plants. So to change anything, you really have to change everything. You've got to go to the top. Fortunately, what's up there, we have some control over. Now, to really understand things, you also have to know what a niche is. A niche, in simple terms, is what an organism needs to flourish. So everything you need to survive and flourish defines your niche. That's the simplest definition. Here we have some dictionary definitions, but I'm going to just stick with everything you need. So as a human being, what defines your niche? Well, let's see. You need food and water and shelter and a mate so you can reproduce. And if you have all those things, then you can continue the species. So that's a simplification of how we can define a niche. And although you have a niche, think about this for a minute, can you also be a niche? This is a representation from an article in Scientific America that demonstrated that we are indeed a niche, and all these different microorganisms that live in us and on us are part of our biology. So we have symbionts too, just like the soil is filled with organisms that work together to increase one another's survival. We have organisms that help us, things in our gut that produce vitamins, things in our skin that just occupy territory, do us no harm, that might prevent other things that are harmful from colonizing our bodies. In fact, you're only actually 10% you. 90% of the cells that you're carrying in and on you are not carrying your DNA. They're other organisms. So we are just as integrated into the environment as everything else. But the message here is that niches are nested. So you have a niche and you create a niche. And this just goes on and on and on. So the more diversity in your ecosystem, the more niches you create. And the more niches you create, the more organisms you can have occupying the same ecosystem. So now let's take a look at the concept of synergism. Now we've got a plant here, and all those purple dots represent the organisms, the community of organisms. There's a lot of different things here that live in and on the roots of this plant. And synergism is when A, B, and C add up to more collectively than they did individually. So let's take a closer look. Let's say we have some organisms in here called A, and they give this plant 3 units of growth. And B gives the planet 2 units of growth, and C gives it 5. You might think, well, I want all 5s, I want all C's, I'm going to get those guys and have really big plants. But when you don't look at them individually and instead look at the benefits collectively, the benefits can be much higher than what they are individually because of all these interactions that go on between organisms, creating more niches, more opportunities to move nutrients and do other beneficial things. So this is synergism, where the sum of the components is exceeded by their combined total. And let's take another example of how this works. So now I have 2 plants. This has one community of organisms associated, and this has a completely different community of organisms associated with it. And when these plants are allowed to interact with each other, they create conditions that allow another community to form based on those interactions. And this is how we get our nesting of those niches and the interactions, building more interactions. And now our diversity is growing as a result of this synergism. So additional interactions can multiply the synergism. And this means that even small increases in plant diversity can yield large increases in microbial diversity. And that's what we're looking for, because it's microbes that are cycling nutrients and keeping things going. Increasing plant diversity increases the complexity of the soil food web, which is responsible for the cycling and availability of nutrients to plants. It's biodiversity that drives ecosystems. That is the take-home message. Agriculture is a simplification of complex food webs, and what you're looking at here is a real food web. And when we subject that land or a similar situation to agricultural practices, the real food web begins to look more like our model. It becomes simplified. We go from a complex field of mixed plants and high diversity to a monoculture. And we lose a lot of interactions that were going on below ground when we do that. And so diversity is lost below ground as well. And the amount of diversity below ground that is lost is larger than you might expect. So the consequence of this simplification of the complex food webs is that you get reduced biodiversity, reduced productivity, and reduced overall system resiliency, because now you're not getting all the benefits that you could have had previously. So let's look a little closer at how reducing complexity reduces resiliency in our cropping systems. I want you to just go back to the beginning. It is the community of AMF that convey multiple benefits to our cropping systems. So now that we've gone from complex to simple, which one of these guys are we gonna be doing without? The guy who protects us from pathogens, the one who's uptaking extra zinc, the one that's keeping the nematodes at bay? Who are we losing? And do we really want to lose any of them? Microbes cycle nutrients, and through their combined actions, they're forming water-stable aggregates in soil. So not only are there direct consequences, but there are indirect consequences. So you need your microbial diversity if you want to have a resilient cropping system. Above ground diversity begets below ground diversity. So if your goal is to create a sustainable and resilient agricultural ecosystem, then to achieve that goal, you need to increase your plant diversity, increase your microbial diversity, improve the drought tolerance of your ecosystem, increase your soil stability so you're keeping your erosion under control, and you want to improve the water holding capacity of the soil. These are the things that are going to make your entire system just function better. And it's kind of all order. So what's the solution to all those things? It's that simple. Cover crops. Remember, you've got to start at the top of the pyramid. That's where you have control, what you plant. Cover crops are an opportunity to introduce plant diversity. This, in turn, increases microbial diversity, plant protection, drought tolerance, nutrient cycling. They increase soil organic matter, which increases your water holding capacity. And a 1% increase in your soil organic matter can gain you 25,000 gallons of water holding capacity. And that's conservative. That's a conservative estimate of how much water you're able to retain. Your soil acts more like a sponge and less like a sieve when you have this organic matter. They physically cover the soil and protect it from the impact of rain and wind and water erosion. And the end result is going to be a stronger, more resilient ecosystem. Okay. Thank you all. I will hand everything.
Thank you, Wendy, for that.
Yeah.
Am I live?
Yes.
Okay. Thank you, Wendy, for that very informative presentation and your amazing pictures. We have a few minutes for some questions, and a few of them have come in. And if you want to send some more in, please type them in the chat box and send them on. I'll give you this one first. Have you seen any production methods that really increase soil microbes that can be used in organic systems? It's tough to do no-till without chemical weed control, isn't it? Michael, you want to take that one first?
Sure. I think the same principles hold. I mean, organic producers are already doing a number of things that are going to help the soil biology. They're not using synthetic inputs. They're using complex organic inputs. So that's a big step in the direction right away. So, my perspective is sometimes you have constraints. You can't do everything, but you try and do the things you have. And organic producers that are keeping the ground covered with a diversity of crops, not putting a lot of inorganic NMP on, are going to be building microbiology. Has been shown. It's been shown in the literature. There are some opportunities. There's a lot of research being done right now on no-till organic systems. And I know one of the scientists at our lab, Randy Anderson, has designed, I think it's a 9-year rotation series for organics that's a no-till and reduces weeds like, 99.9%. But there's other people. I went to a national meeting a few weeks ago, and it looks like it's a fairly active topic right now.
Here's a question for Dr. Tahari. Are there cover crops that benefit AMF more than others?
There are. We have been doing some of that research, Mike and I, before I separated from the USDA, and we have found that the amount of diversity in different cover crops does vary. For instance, if you compared hairy vetch to oats, we found that everything that all the different species and genera that were present in vetch were also included in oats, plus oats had, 5, 6 different additional species that they also harbored inside their roots. So they are not all created equal as far as how much diversity the different cover crops, the different species promote, and we don't have a lot of research yet on exactly which cover crops promote which species the best. So that is a growing area of research. And at the same time, if you shoot for, until there's more information, just increasing your diversity, maximizing it, get as many different crop species as you can into your cover crop mix, you're going to be covering your bases pretty well.
Mm-hmm. And carrying on to that one, does depth of tillage make a difference to the AMF populations?
Yes, obviously the deeper you go, the more damage you're going to do to those filamentous hyphal networks that are under the soil. And they have a purpose. When new spores each year germinate and colonize plants, the first thing they do is race down and through that existing network from the year before, reinforcing it, reabsorbing it. And so it's a big time saver to how quickly they can get to cycling nutrients. If it's not there because you've tilled, then it takes them much longer to reestablish. Plus, they're going to need more carbon from the plant to help them reestablish that network.
Thank you. I'll give this one to Dr. Lehman. Has there been any research to show how tiled fields are affected by cover crops? Would cover crops negatively affect the drainage or filtration on a tiled field?
Right off the top of my head, I'm not thinking of any research that examined the interaction between cover crops and tile drainage and how that might affect soil microbes. I guess I'm just not thinking of any off the top of my head.
Mm-hmm. Okay. Here's a grazing question. Any research on how much grazing cover crops might benefit AMF?
How much grazing? Well, I personally like Gabe Brown's model where they go in and graze half of it off. One of the things that you lose when you don't have cattle integrated into your system are the microbes that come out the back end of the cattle. And a lot of those microbes play a role in cycling phosphorus and other nutrients. And so getting that natural inoculant back into the ground, I think, is very important.
And building off that natural inoculant, here's a question just on that. What about AMF inoculants? Are they needed, and if so, when?
Yes, they're needed, but I think the industry has a long way to go before they have inoculants that really work. Most inoculants are based on only a few species. There's only a dozen species out there, and as you can see, we need a lot more diversity than that. And often the inoculants are dominated by a single species. They might be mostly rhizophagous, which I usually see in most soils anyway, because they've adapted well to the agricultural atmosphere and climate and the high inputs, and they've managed to hang on where a lot of other things have disappeared. So I would say we need more research into that area, and that the benefits are probably limited to really degraded soils right now until we have more research and more knowledge on that.
Okay. Here's a question for Dr. Lehman. What factors determine the percent of total fixed carbon that are exuded from the plant into the rooting zone?
Good question. I'm certain that it varies according to plant type and to environmental conditions. I'm afraid I can't provide you generalizations. I mean, that's a range that's reported in the literature. If you look at a single study that uses a certain plant, they'll generally fall in that range, but I have not synthesized that information that you're after.
Okay. Here's a question about amazing pictures and information. How do we convince folks that ecology is a real science?
Well, wow. I didn't know that people didn't realize it was a real science, so that's a tough one. I don't know.
Okay. Here's a question. What can I say to farmers that know this but still believe a black field means warmer soil in the spring and earlier planting? Either one of you?
Did you want me to take that, Wendy?
Yeah, go ahead, Mike.
I think in some parts of the country, that's probably a reality. The default, I mean, I heard today from some guys, there's some folks that live that work here at the lab, that parents farm not too far away, and it came down to Dad likes to drive the tractor, and it's a sense of accomplishment, and so there is that, and it's going to take a long time to sort of relearn that. I realize there are certain locations that have specific environmental conditions, and we're trying to grow something that maybe is not best suited for that place, but if you own it and you want to make it profitable, that's what you're going to grow. So we do have some of those issues to work out, and I think that's an area where things like programs and incentives come in to to help you do that, because in the long run, if you are cover cropping, if you are keeping the ground covered, you will have better soil structure. Better soil structure means better aeration, better infiltration, less erosion, more water storage, able to support heavy equipment better, resist compaction, all these positive things. And if you have all these positive things going, you've got a lot of soil microbes in there, and guess what? They make heat. They actually can raise the temperature simply because of their metabolic activities. Anybody who's seen a big compost heap smoking away in the winter can attest to that. So, again, I think it's a question of balancing priorities, but more importantly, I think it's the time scale. Are we looking at an annual time scale, or are we looking at something longer?
Okay, I think this one is for Dr. Lehman. Are there ratios of fungi, bacteria, protozoa that we should be aiming for in soils?
I don't believe so. Some folks will say yes, definitely. My feeling is that the fungal bacterial ratio is very helpful. It's not like a ruler. I mean, the lines on that ruler are fixed, and where with fungal bacterial ratios, it's a guide, and it's method dependent. There's at least four ways to measure fungal bacterial ratios, and the actual numbers you get will be different. I think it's very useful for relative comparisons between two treatments, and also for over time, which direction are you going? In my mind, that's the utility of fungal bacterial ratios and not necessarily a magic number.
What is the additional benefit of cattle eating the cover crops on row crop fields?
Well, for one, it can help with crop termination. Two, it is a low-cost fodder. The carbon goes back into the soil because they don't fully digest everything. And also, the microbes in their gut have been found to produce enzyme called phytase, which helps free carbon and work in restoring the carbon cycle, so that when you get your soil tested, you're mostly getting back your inorganic carbon, things that they call available carbon, but the organic side is kind of ignored because it's a complex process. And if we want to keep the cycle working, we need to take that organic portion of the cycle into account, and that's where phytase comes in, freeing up organic carbon, helping it become mineralized into an available form. So they play a variety of roles there, plus they're pretty tasty.
I'd like to add a couple things on that. If you directly graze your cattle, I would think from producer standpoint, you're not paying a lot of transportation costs, expenses, diesel, etc., to move bales or move material to another location and then try and move the manure back. But I think the biggest improvement would be the equitable spreading of the manure in so much as you can over the field. So I think if all the cattle are in one corner of the field, that maybe it's not as good as if you can get it dispersed out over the field.
And I could add one more thing to that, and our entomologist isn't here today, but we work closely with Jonathan Lundgren, and one of the things that we're seeing is the loss of dung beetles. And if you want to incorporate your fertilizer into the soil, what better way than to have the beetles do it for you? No diesel fuel is spent, you're not making more passes and compacting your fields, and we're finding that they're declining because we've really separated out our animal and plant production systems, and at the same time a lot of the chemicals that we use in these systems are also harming them.
Okay, we're getting down towards the end of the time, but for those of us who are new to this, could you briefly go over the timetable for cover cropping? For example, do you plant the crop one year and then till it under the following year?
Mike, I'll let you have that one.
Oh, well, let's see. My mic is on. I am not an agronomist. I'm still a microbiologist. On our farm, our research farm, we don't have any livestock, unfortunately, and largely because it's a huge expense for the research station. So we have our cover crops. We used to just plant them into a 3-year rotation after the small grain phase. Now we've got some adapted instruments that allow you to interseed the cover crop during corn or soybean, like say V6, somewhere around there. So that's how we're doing the cover crops. They're generally chemically terminated in the spring, roughly around the time of seeding, sometimes actually after the seeding of the next year's crop. And so we don't have cattle integrated into our cover crop systems at our research station.
Okay. I think this will be our last question. What are the very best ways to get effective cover crops in corn, beans, rotations up here in the Northland?
Well, I just went to a little, well, it was more than a little, it was about 200 folks there in Okabena last week. And that was the main theme of that conference, was how to get the cover crops. And you know, there was some folks flying it on, and they were discussing the economics of doing that and what things to do to make the economics more favorable, like pooling resources with neighboring farmers and innovative ways to keep the seed bin full on the plane, you know, and minimize that downtime that you're paying for when the plane's not actually applying seed. You know, you can apply with a high boy. I saw some pictures, and I don't remember who had it, it was a really super high boy type thing that appeared to be air seeding the cover crop. And then there are a couple folks out there that have modified some machines to actually drill the cover crops in between the rows, you know, at say V6. So this is a really rapidly moving area where people are now starting to put their attention. At our farm, over the years, when we've tried to broadcast seed them, you know, if it doesn't rain, not much happens. But what we think is that even a little bit of cover crop is good. Even just a small plant, if that's all you get, that's good. We've tried short season varieties so you can direct seed afterwards, but we've got a reduced yield from that approach, and so we think some manner of direct seeding, or if you could make the economics work and figure out when it's going to rain, you could broadcast it.
Okay, well, I think we've used up all our time. This is very, very good information and good discussions on the questions and the answers. Thanks again for attending this webinar, and thank you to Dr. Lehman and Dr. Tahiri for your presentations. And again, a reminder, as a way to help us determine if this webinar was useful to you and to help us plan future education events, please reply to the short survey that will be coming soon. And again, I remind you to further your knowledge of soil health and cover crops by registering and attending the Midwest Soil Health Summit in February. Hope to see you there. Thank you, everyone. Goodbye.